So just a word about the motivation, and Philip already mentioned some of those elements this morning. So microbial activity will be essential to all soils. So even if you do not work directly with microorganisms, there's a good chance that they actually somehow also affect your work directly or indirectly. I'm thinking in particular about soil formation and soil structure, or they can affect those in non-trivial ways that could be also of interest for you. So here's the program for today. I'm just going to introduce briefly soil microorganisms, given a little bit of context. For the sake of time today, I'm going to skip out a part on biogeography of soil microbes. I'm going to skip this, but this will be in the PDF that will be available after the summer school. I'm going to talk more about soil as a microbial habitat, and in particular, what are the consequences for different microbial processes, dispersal, activity, and spatial organization. And if I still have time, I'll finish with a few elements about soil formation, structure, and physical properties. I also decided to add just a few technical notes that will be just basic information about soil biology, soil microbiology, just to make sure that we're on the same page, that we have some of the fundamental concepts there. So most of the material that I'm presenting today is actually directly coming from this review paper that Danny and I published last year in FEMS Microbiology Reviews. Feel free, if you want to know more, to look at it. It's open access and it's available online. So let's jump right in about soil microorganisms. So what do we talk about when we talk about soil microorganisms? We talk about a vast diversity of organisms. So you'll find really different types of cells. We can find, for instance, bacteria and archaea that are really the simplest unicellular organisms that are called prokaryotes because they do not have a nucleus. They are the simplest and ancient forms of life. But those they are pretty abundant, and you can have more than 5 tons of those prokaryotes in an hectare of soil. And then we have a lot of eukaryotes, so that means all the cells that possess a nucleus, and you'll find algae, fungi, that will be really important. You can have up to 15 tons of fungi per hectare. Protozoans, slime molds, which are weird creatures that are somewhere in between fungi and protozoans. Just to give you this overview. And last but not least, we have viruses. Viruses are not per se alive, but they are very abundant in soil, and they obviously also play an important role. There's a high abundance of those microorganisms out there. If you think of, if you take 1 gram of maybe topsoil, you can have tens of billions of bacteria and archaea, hundreds of thousands of protists, so protozoa, algae, slime molds, maybe 100 meters of fungal hyphae, billions of viruses, and you also have an equal diversity, and the highest estimates for bacterial diversity range from 1,000 to 1 million phylotypes, so that would be not directly a species, but you could think of it a little bit in that way per gram of soil, so really amazing diversity. So let me just show you some images, some videos of the type of microbes that we isolated this week in Cargèse. So just to show you here, the bottom here are some soil bacteria that we grew from the soil on a selected medium. And then we had a group also enriching bacteria from sand, from the beach. And what you see here on those images are a lot of small black dots, so there are bacteria swimming around. And because there's a lot of bacteria, they have a lot of predators. So we also see those ciliates here who feed on bacteria and that are very happy to grow as we also boosted the bacterial growth. So just some images that we took last week during the workshop. Give you an idea, the bacteria typically will be just a few micrometer long. The protozoans here are more like tens of microns long. This one is up to 100 micrometer long. Okay, this is to also say that there's a range of sizes in those soil microbes. There's an interesting size-density relationship here. So on that axis, you have the density, how many individuals per square meter as function of their size. And here are our microbes, so we have the bacteria, fungi, algae, protozoa. You can see there's quite some variety also in the size range. But the important point is that the bacteria are really the most abundant one in soils. Bacteria are much smaller than fungal hyphae usually, so you see here a scanning electron microscope of a fungal hyphae that is colonized by bacteria. This is the same type of situation, but with a fluorescence microscopy image. So bacteria and filamentous fungi, which are really the big player in soil, they can be really different in terms of morphology, physiology, and more. So again, bacteria are the most abundant ones, and for that reason, I'm going to talk mostly about bacteria during this lecture, but I'm going to also mention fungi whenever appropriate. Here's, let's say, the way a microbiologist see life. This is a tree of life, relatively recent. So you don't have to try to read the details. Here's the big picture. You have all branches that show different lineages, different species of bacteria that are connected. And the way we microbiologists look at life is there's three main domains. We have the bacteria, we have the archaea. Again, those are the simplest unicellular organisms. They look the same, but they are quite different. And then you have the eukarya, that's the rest. So in that small branch, you get the fungi, the plants, the animals, the insect, us. We're all in this small branch that's not that really interesting, actually, in terms of diversity. Most of the diversity is here in the bacteria. What is really amazing is that most of this bacterial diversity is known only through molecular signatures. That means we've isolated DNA from soil, we've sequenced it, and we found out that this DNA belonged to species of bacteria. The red dots indicate lineages that don't have an isolated representative. That means that we are not able to grow those things in the lab. We know them only through the DNA. And as you can see, there are many red dots here. That means that a lot of the diversity is not known. We can use this DNA information to compare sequences. We use similarity of DNA sequences to build trees, such as this one. This is called a phylogenetic tree, that show the different relationship between the organisms. Yeah.
Is there a reason why there is much more diversity in bacteria than in eukarya?
Well, one element is that first they are much more ancient, so there's also more time to accumulate diversity. And then the diversity here is really in terms of also of the sequences. You could have sometimes, for instance, in eukaryotes, you can have sequences that are very close, but at your scale, you would consider that there's a big difference. So, for instance, a sequence similarity that we have with chimps is pretty low, but I'm sure you would agree that's kind of a different kind of organism. But here we're looking at much larger differences, and the fact that they are very ancient is also an explanation for this. And here I'm just going to introduce my first technical note, which is about how we can use certain molecules and the sequence of those molecules to predict some relationship and use them as evolutionary chronometer. So that's a little cartoon showing a bacterium where it's had its DNA, so it doesn't have a nucleus, it just has a chromosome in the center. And each cell contains hundreds of thousands of those little elements here called ribosomes. So ribosomes are essential to all cells because the job of the ribosome in the cell is to read the information from the DNA, the information coded in the DNA. It has the form of a so-called messenger RNA, and it translates this information into amino acid chains that form proteins. So all cells, all living cells have ribosomes, so they are very well conserved. And what is important is that a part of those ribosomes is made of RNA, and the several regions of those RNA are really well conserved and can be used as evolutionary chronometers. So we can read those sequences, and we can then say something about the relationship between organisms. If you start reading about soil microbiology, you will often see mentions of the so-called small subunit or 16S ribosomal RNA. This is the most commonly used for the classification of bacteria. And as of today, the Ribosomal Database Project, which is a freely available database, contains more than 3 million bacterial RNA sequences and more than 100,000 fungal sequences. So with this, you can just go out there, sequence the DNA from your soil, and try to match it to the database, and you can get already an idea of what you have, so you don't need to regrow everything. And with this information, you can also build complex trees. Oh, sorry.
So for me, a virus is a bunch of DNA as well. And
yeah.
How can you distinguish between a bacteria and the virus RNA and stuff like this?
Yes. So the virus, in that particular case, the virus don't have ribosomes, and they don't have the particular genes that would be there. But typically what the viruses do is they hijack a cell, so they use the machinery of cell to replicate. But actually, what you can do with ribosomes, you can do with other genes. If you read DNA, if you compare it to the database, most probably you will find a match with a virus, if it's a virus we're interested in. And if the match is pretty good, then you will have a very you'll be very confident that it's actually a virus that you targeted or isolated. But you can select different genes. We used here ribosomal RNA. Earlier, people had used directly the amino acid sequence of proteins to compare. So that was pioneered in I think the 50s or 60s by Linus Pauling, and he had really this great insight that we can use molecules to predict what is the evolutionary relationship between organisms. So again, here we'll talk again a little bit about 16S ribosomal RNA, a convenient way of relating microbes with each other. So now a few general things about soil bacteria. First, there's really some recent research. It's interesting that as recent as this year in Science, looking at the diversity at the global scale. And here, again, this is using this information that is directly coming from the DNA, so you don't have to regrow the organisms. It shows that some are very abundant. Those are examples of very abundant soil microorganisms. And others can be present, but only very rare. And that is often the case that a lot of the diversity in soil microbes is actually in populations that are very small. So some are very abundant, some are very rare, but they all participate to the diversity. And something interesting about soil bacteria is that they tend to have larger genomes and to have more genes than the bacteria that come from aquatic environments, for instance. So here on that graph, what you see, I've plotted data from the US Joint Genome Institute. So more than 1,000 whole genomes, so full genomes from individual strains and organisms. And on that axis, you have the genome size, megabase. On that axis, you have the number of genes. And then those are the environments where the isolate came from. And what you'll see is that soil and the rhizoplane, so bacteria that are associated plant roots, are the ones that tend to have the largest genome and number of genes. So you can also see that there's very good correlation between the number of genes and the genome size. That is typical of soil bacteria, because bacteria don't make waste. They really pack their genome with genes. If you do this with animals or plants, you don't find this picture. Just to realize, some of those bacteria has as many as 10,000 genes. Human beings, usually we estimate that we have about 20,000 genes, so 2 times more. But we have a genome that is about 3,000 megabase. So we have a vast discrepancy between the number of genes and the genome size. A lot of a human genome is just junk. It's just accumulated stuff that have been with us for a long time, but that don't necessarily play a role. So a lot of those genes are so-called accessory genes. That means they are not essential to the bacteria, but they help them in specific tasks. For instance, it helps them sense their environment, transport molecules, produce antibiotics. As a side note, I think it's important to know that more than 80% of the antibiotics that are currently used in hospitals originate from soil bacteria, such as streptomyces here. It's really a huge contribution of soil microbes to our own well-being. Also, it's important to know that the genes that you have matter, but also what you do with them matter. If you express them or not can vary a lot, depending on what environment you live in. And there's a lot of metabolic variability, and we also saw some examples this morning with Philip. There's really a tremendous diversity of metabolism in the microbial world. So that's it for this brief overview, and we're going to jump into the environment and talk more about the physical aspects of soil as a microbial habitat. So again, talking more about bacteria. Bacteria, they inhabit the pore spaces and the soil grain surfaces. This is a fluorescence microscope image of a soil thin section where the little blue dots are actually bacteria that were stained with the fluorescent dyes. And you can see that they will cover the surfaces of the pores in the soil where they can attach to the particles. Or here you can see a scanning electron microscope image of some bacteria that we isolated from a forest soil in Zurich. So when you look at them at the bulk soil, you will find high densities of bacteria. as I mentioned before. But actually, it's estimated that far less than 1% of all the available soil surfaces are occupied by microbes. And also, depending on the soil type, you can have 15 to 50% of the soil porosity that is not accessible to the microbes due to the size of the pore folds. And finally, but maybe most importantly, the bacteria, they need an aquatic environment if they have to function. And in soil, the water phase is often fragmented and dynamic. And we're going to talk more about the availability of the water phase because it is really an essential element for soil microorganisms. So if you use a little cartoon like this, you can imagine, conceptualize that water is retained by the capillary forces in corners and crevices. It can be also absorbed as a thin water film to the surface of soil grain. But interestingly, if you try to calculate what are the film thicknesses that you find, you can use simple assumptions. So for instance, if we assume that only van der Waals forces are playing a role here, there's a way to calculate what is the effective film thickness, water film thickness, as function of the metric potential. And the bottom line here is that if you use values of metric potential near the field capacity, it tells you that the film should be about 10 to 20 nanometer thick. Now, a typical bacterial cell is more like 0.5 to 1 micrometer wide. So it is way smaller than the typical bacterial cell size. So they are still connected to liquid if they're there. There might be nutrients diffusing, but they are pretty much immobilized because it's as if you want to swim, but you're on the beach there and you just have your feet under the water and you're like, here, you're not going to go anywhere by swimming, but you're still connected to the medium around you. But now it's a bit different if you start looking at what's happening in the pores or crevices. Here, this is also connected to what Danny presented last week. You can use the Young-Laplace equation to estimate this radius here, depending on the metric potential and the water surface tension. And then, if you assume certain angles, you can try to predict what is the radius of a cell that could be immersed here in this water. And there, if you say, okay, angle 30 degrees, again, field capacity for the metric potential, then we find a radius that's much closer to what we expect bacteria to be, so 0.8 micrometer. So it tells you that you're more likely to find those in those kind of areas that adsorbed to a surface. And this is also showed in this graph where we have both model results and empirical results of what is the effective film thickness here and as a function of the metric potential here. And if you assume a perfectly flat surface, you'll see that the film is always very thin here. Maybe you cannot read, but this is 10 to minus 7 meters, and that is 100 nanometer. And if you have a rough surface, then it jumps to something much larger, so a tenth of a millimeter. So you really have orders of magnitude differences depending on the roughness of the surface. And of course, this will really play a role on rough soil surfaces that can carry and support those microbes that are there. So this is at the very small scale, but you can also consider this problematic of water at a larger scale, such as the soil profile scale, where in this little cartoon we just represented a soil profile with decreasing concentration of oxygen and carbon as you move down in the soil profile. And just imagine that some of the time in soil you're going to have wet events, you're going to have rainfall or irrigation. And then this temporarily the water content will increase drastically in soil, and maybe we'll find even saturated conditions for the microorganisms. And as the soil dries, as the water evaporates or is taken over by the plant roots, over a couple of days we'll go back to unsaturated conditions, and bacteria will now be exposed to a two-phase environment with liquid and also air in there. And what is interesting is that in most climatic and geographic regions, and most of the times we expect that the soil will be more like this than like this. But they will be exposed wherever they are to those rapid changes in water potential and oxygen concentration. And of course, this has consequences for their dispersion for their activity in soil. Yeah?
Why you have the similar profile of carbon? Because you consider that the organic matter is exactly...
Yeah, so the reason here, we're just imagining like a situation where relatively rapid changes and no necessarily activity. You expect that if you have water, you're going to change the water and oxygen profile, but not directly the carbon. It's true, yes, you could imagine that some of it can be moved around, but in that case, to simplify, let's say that most of it does not change as fast as the water and oxygen. We have the different in time scale here mostly. Over longer periods, you might have changes. Another important thing when you think about microbial activity in soil is the fact that the carbon that is out there is not homogeneously distributed. You'll have, it's going to be heterogeneous in space and time. And because of that, we also have good estimates. We know that only a few % of the soil microbes are active at a given time. The majority is inactive or dormant. So microbial abundance and activity is associated with what we call hotspots. That are local areas where we have increased microbial abundance and activity. And those hotspots could be, for instance, bacteria colonizing a root hair tip. There will be some extra mucilage to be eaten, and there will be some goodies secreted by the roots. Or that could be maybe some decaying plant material further down in the soil profile. But here, I just added those little red dots. Imagine that if you look at microbial activity, it's not going to be homogeneous in the soil profile. You're going to find hotspots here or there, depending on some additional factors, and those hotspots probably change over time. Okay. So with this, I think now we move to, to more precisely, what are the consequences for important processes such as dispersal, activity, and spatial organization. And I want to start with dispersal of microbes. Here I took this nice illustration from a good review by Voss et al., where they look at the variety of dispersal mechanisms. And here it's important to realize that those mechanisms operate at a range of spatial and temporal scales. So you have really small scale, small time scale processes, cell division, motility, up to global scale processes, maybe dispersion by wind, maybe transport even by humans if they move soil around. And we have really these vast scales of processes that all can play a role in the end in the activity of the microorganisms. The dispersal can be active, so when we talk about microbes, we talk about motility, or that can be passive through the water flow or transports by animals or wind. Bacteria can be transported by earthworms, by other small inhabitants of the soil. So you always have to think that there are active and passive processes. What is interesting is that when you think of the advection of soil microbes, so it's facilitated by streams of water. But what is expected is that, with a few exceptions, in most unsaturated soil, most of the times, the condition will not support advection. And here are some modeling results from my colleague Ali Ibrahimi, who used a simple model of bacterial movement in idealized pore networks to ask what is the ratio between passive and active transport. In that case was the ratio between convective capillary flow and chemotactic motion. Chemotactic motion is just to say that the bacteria do not move randomly, but they are attracted, for example, by a certain source of nutrients, and they direct their movement towards a gradient. And what it did that for 2 diameters of cells, so 1 micrometer or 2.5 micrometer. And what you see is that as function of metric potential, very rapidly, this ratio becomes very, very small. That means that it's dominated by the chemotactic motion. So that's important to say that it is when the soil is unsaturated. Of course, you will have episodes of saturation after rainfall or so. And it could be that those episodes, even if they are relatively brief, that they will be also fundamental for the dispersal of the organisms. Doesn't mean that because most of the time it's unsaturated, you don't have important transport under saturated conditions. And another thing, in addition to this, something that's been discovered only a few years ago, but which I think is very important, is that bacteria can also, oh, sorry, I forgot about this one, move, let me see, no, can also move, and I will talk about this later, can also move, also transported by animals, or move also along fungal hyphae, and I'll show this later. But now I will just say a few words about the many ways microbes move, another technical note, just for that you get an idea of what are the mechanisms there. So soil microorganisms can move thanks to rotating or moving appendages. So in bacteria, this is soil bacterium, Pseudomonas putida. It has appendages called flagella. There are nice little appendages can be rotating, and then it propels the cell. In ciliate, for instance, we have those are larger organisms. They are not at the same scale. We have other types of appendages named cilia. Typically, those organisms, if they are immersed in a water film, they're able to swim, and that is a very useful way for them to disperse in their environment. But it's important to know that there are many other types of mechanisms to disperse. When they are on surfaces, on hydrated surfaces, bacteria can do other things called swarming, which is a communal way of moving around. Things called twitching, gliding, sliding, whatever. Many different things. So there's not just one mechanism to move around. There are many ways bacteria can do this. And importantly, gradient-guided swimming is commonplace. That means that when you're able to swim, in most cases, you're also able to orientate in your environments. There are places you want to go, there are places you want to avoid, and most bacteria, they have very refined sensing machineries to do this. They can sense, let's say, a glucose gradient, or they can detect like a chemical that is toxic to them, and they want to swim away from it. So they have ways of orientating their swimming. So now, a few more about how the metric potential controls this motility. The way you can look at this is you take some bacteria that are swimming in the soil. So as soil dries, you'll see that the water film will become thinner and thinner until at some point the cells are just trapped in a very thin water film. They are pinned down to the surface by the very strong capillary forces. And again, this is a way of simplifying this view, and you can what you can do is you can try to express their velocity as a function of their self-propulsion, so they can move their flagella and swim. And then that is hindered by the cell surface interaction and by the capillary pinning force. And what we did, not directly in soil, but on models systems, both in simulations and in the lab, we're able to measure this velocity here. So the measured data are the points here, and this is as function of metric potential. And we also compared with different models. And you could see that you have this decrease of velocity as function of metric potential. So you have a direct control of the physical environment on their ability to swim. But maybe more important than the velocity per se, because there's a decrease, but and until they're pinned down, they will not move at all. But the decrease is not necessarily that important. From 15 to 5 micrometer per sec, it's not huge. But what we think is more important is that as the soil dries, you sort of create fragmented habitats, fragmented aquatic habitats for the microorganisms. And this is what we also tested with a soil bacterium, Pseudomonas protegens, trying to look at its dispersal on a model rough surface. We were just using here porous ceramic surfaces as a model. And if you start in a pretty wet environment, you can look at all those bacteria swim. And this picture here shows you little dots is the path taken by a swimming bacterium. So we can record it over a few seconds and just project its swimming trajectory. And here the radius shows you how far it can go for a given amount of time. And if you dry this surface just slightly, I mean, it's very, very mild metric potential, it already creates those fragmented aquatic habitats. And you can see that now the cells are trapped in a small area, and they can swim. They cannot really escape it because they are limited by the thickness of the water film that they can swim through. And this is just a measurement of this. If you are at 0 kilopascal, you can go further than at already a very mild metric potential. So flagellar motility in soil seems to be restricted to really a narrow range of very wet conditions, because you see already at -2 kilopascal, it doesn't move that much. Although there are ways around it. So again, if it's, even if those wetting events are short-lasting, that doesn't mean that they don't play a big role in the ecology of those microorganisms. If they just need to be taking the best of those events, they have a few minutes maybe to swim, to colonize new environments, and they can do this. And know that, yeah, sorry.
In these thin films, are they still moving along gradients? Or does the gradient in water film thickness dominate the direction they move?
Sorry, but first.
On the previous slide, you said that these bacteria moved driven by gradient.
Yeah.
Of nutrients or something else.
Yes.
Is that still the case in these thin films? Or does the gradient in water thickness dominate the direction they move?
No, they won't. Yeah, in those cases, even if they would like to go someplace, they will be hindered by the thin water film. So they could still sense the gradient, but they won't be able to use that information efficiently to go there, or at least not on a short time scale. And the other thing that I was mentioning this before is transport by fungal hyphae, and I think this is a very interesting additional idea about how bacteria can move around in soil. The idea is that these structures made by fungal can be used as so-called highways by bacteria, and that can really increase their dispersion up to 1 centimeter per day in the lab. And of course, that depends on, for instance, if the bacteria are expressing flagella. Not all bacteria can swim. And what are the hydrophilic properties of the fungi in the bacteria. And here I'm just going to show you a movie made by Tom Berthold at the Umweltforschungszentrum in Leipzig, where he directly observed this movement. Let me find the video. It's on YouTube if you're interested. And what is nice is that it's really shown in real time. So they use just one species of bacteria and one species of fungi. And what you see here, so those fungal hyphae, they're about 10 micrometer wide probably. And the movement that you see is those bacteria that are sort of, driving on that highway, going places. Here it's on a simple environment, but just imagine for a moment that there are hundreds of meters of hyphae, maybe in a gram of soil, so they're all like routes that the bacteria can use to go places, even when the conditions are not necessarily fully saturated. So you see this really beautiful network of mycelium there. So you see the scale here, 20 micrometers. A fungal hyphae is about probably 10 in the distances. In that case, all the bacteria are just a few micrometer wide. Yeah.
Their movement is driven by chemotaxis. Why are they going into opposite directions?
Well, they might be responding to chemotaxis, but even yeah, those are references. Even not, there will also be some random swimming at some point. But on those hyphae, what they've done also is experiments where, and we don't need the next video. So they have used those experiments to show that bacteria swimming on hyphae could still sense gradient, and then they could attract them. They could use the hyphae to go to a new environment that would contain, let's say, an attractant, and that they would go there. So they would still be able to sense gradient, but it's also slightly more complicated to see. Okay, so many ways to move around in soil, and really fungal hyphae. Of course, this was done in the lab, but I'm absolutely convinced that it's also taking place in natural soils. Now let's go back to microbial activity and particularly microbial respiration. And now on that graph, I'm showing you several things. There's in red, there's theoretical soil relative humidity here as function of the soil water potential. And the little gray dots here are data of microbial respiration rates were normalized to the value at field capacity. And I have to thank Stefano Manzoni who gave us those results here. A few interesting things. So when you look, there's this very nice decrease of the respiration as you decrease the soil water potential. But you don't see that clear with the relative humidity. So the soil remains pretty humid. Relative humidity is high throughout the long, across many values of soil water potential. Here, just to give you some ideas also, I've put additional keystone values here. For instance, around that point, you don't have any flagellum motility possible anymore. Around that point, you don't have any bacterial respiration, more or less. That means that a lot of the respiration that goes further down is probably due to fungi. And what is interesting is that if you compare to the soil water potential, the relative humidity really has little influence on the activity. And to me, that was kind of a surprise. So almost all the activity takes place between 90 and 100% relative humidity. So with this, what the message is really, if you want to look at activity, you want to look into soil water potential. Relative humidity will not tell you much. Now, what are the effects of this metric potential on aqueous and gaseous diffusion? So here I'm going to show you some results again from my colleague Ali Ibrahimi, where I used a 3D pore network, a mathematical model to predict what would be the relative diffusivity of gas and solutes in a porous environment such as found in soil. What is interesting is that as you dry your porous environment, you will reduce the liquid pathways, and then you will have lower relative aqueous diffusion rates. It's easy to understand that if you have in a pore network like this, if you have less and less liquid, it's like a maze. At some point, the molecule still travel at its diffusion constant, but it might have to find a longer path to the other side. So the relative diffusivity changes. And what Ali showed is that you have this decrease here of the relative diffusivity of an aqueous environment as function of the metric potential. And this was in pretty good agreement with empirical model from the 60s. Now you have the other side of the coin, which is that as soil dries, then gaseous diffusion rates increases. For instance, oxygen diffuses, and the medium becomes aerated. So this is gaseous diffusion here. So again, this time you have the opposite picture. As soil dries, metric potential decreases here, and you have this jump here in gaseous diffusion. Because of course, oxygen diffuses much faster in the air than in water. So it's about, I think, 10,000 times faster. So from that picture, then comes an idea, is that possibly this interplay of aqueous and gaseous diffusion could lead to an optimal water content for microbial activity at the larger scale. And that was proposed by Scott already a few decades ago. The conceptual view is that because you have substrate diffusion on the one hand and oxygen diffusion on the other hand, maybe there's an ideal water content where you're going to get the best of both worlds. You're going to get your solutes diffusing to you, and you're going to get oxygen that, as we learned this morning, is the best oxidants that you want to use to grow. And interestingly, there's not a lot, but there's a few evidence for this optimal water content, again from Scott, looking at soil respiration here as function of relative water content. You have this sort of peak of respiration. There were some other experiments in sand microcosms. I think in that case it was only using one species of bacteria, looking at O2 consumption. We have also this little peak here. Other measurement of oxygen consumption in different studies. So there's a few evidence that this might be important here. Now this is about aerobic respiration, when you use oxygen to burn sugars or other type of organic material. But of course there are other types of respiration. We have anaerobic processes in soil, and I'm going to also spend a bit of time on that because it's quite important. We have anaerobic respiration that can take place, and this is the job of so-called facultative anaerobes. For instance, they can use nitrate or nitrite as electron acceptors. And they will then produce nitrous oxide or nitrogen gas. And this is this process known as denitrification. And here again, some modeling results where in that model, again, there was a pore network model that was used with populated with two species. We have a green species, which is an anaerobic species, can use nitrates to respire, and a blue species, which is aerobic. And here, interestingly, if you vary metric potential, and in such models, each single microbe is represented as an individual. So the little dots that you see dancing here are individual microbes which are moving in the pores, consuming nutrients, growing, dividing, and so forth. And what you see is that as a function of metric potential, you will have, we'll find our optimal peak for the aerobes, was expected. But you also find that when it's pretty saturated, when there's low oxygen content, you have now the anaerobes that are thriving. And the organization in space matters because you will find those anaerobes to the center, because in such models, the idea is that the oxygen diffuses from the outside of this little pore network. The idea here was to somehow mimic what you would find in a soil aggregate. Let's say you have a little bit of organic matter in the center. You have pores, saturated pores, and you have oxygen coming from the outside. So closer to the outside, you're going to get aerobic processes. Closer to the inside, you're going to get anaerobic processes. What is quite important is that, again, I'll come back to this concept of hotspots. We might have 1% of soil volume, which account for up to 90% of the denitrification activity. So you have small elements that contribute massively to some processes in soil. And again, local anoxic conditions can persist even in aerated soils. So for instance, in an aggregate like this, and this I will also discuss in the next slide. We talked about facultative anaerobes. We also have obligate anaerobes. Those are groups like Clostridia, sulfate-reducing bacteria that we saw earlier, Methanogens. And that is also a key thing, because if you look at methanogens, so those that produce methane out of CO2, they produce almost all the biogenic methane on Earth. And what is interesting is that usually they cannot grow if there's air. They die in presence of air. But despite this fact, they are almost ubiquitous in soils, including all unsaturated soils. There was an interesting experiment made by those gentlemen here. They took an arid soil from South Africa that had been stored dry for 10 years probably forgotten somewhere in a lab. And then they took that and they re-wetted it and incubated it, and they were able to detect methagenic activity. So for years it was kept fully aerated, but still you could revive the microbes that were in there that were able to produce methane. And more recently here from Angel et al., we saw that those archaea, so methanogens are always archaea, are globally ubiquitous. And here you use also this technique of looking for the 16S RNA gene. You can measure the number of genes that are in your soil, and that gives you an idea of how many cells were there. And you see that this value correlates pretty well with the methane production rate that was measured, for instance, in those soil cores. And they looked for one particular species here, or group of species. Okay. Just a final technical note, because I find it fast. The story about the methanogens, that they are everywhere, but they're not necessarily active. So it's clear that the majority of soil microbes are not active. And this is an interesting representation from Lennon and Jones, showing the percentage of inactive cells in different environments. Here's soil, 80%. It's an estimate, but look at compared to human gut, 20%, or marine water, etc. So that the majority of bacteria and microbes are not active. So they coined this term of seed bank. You have a reservoir of dormant individuals that can potentially be resuscitated in the future under different environmental conditions. So what you have at a certain time point is not necessarily what you can get the next day if you change the conditions. They do this because they enter what is called dormancy, and that can include the formation of spores. This is a micrograph showing such spores. Those are the white, bright objects there. Typically, Bacilli and Clostridia do spores. Other actinobacteria and filamentous fungi do other types of spores. Those spores can persist for years. I mean, they found samples that where spores were maybe thousands of years old, and they could still resuscitate them. So this is this gigantic seed bank that exists in soil, and that can be mobilized or if the conditions change. Just a word now about species coexistence and diversity. Here, most of the information comes from microcosm experiments, and there's some interesting idea. For instance, that the spatial isolation of the microbes, due to the changes in water content, will have an influence of their coexistence and diversity. Here, an experiment where they looked at log CFU, just a number of bacteria, huge, and they had just 2 species, the white species here, gray species, as function of metric potential. Under wet conditions, they found only 1. That means that the other was excluded by competition. But as the soil dried, they found both. And we found something more recently, something similar with different species, also again in sand microcosms. Under wet conditions, you see the ratio between the black and the white. And this is a log scale, so the ratio is pretty high. And under dry conditions, now they are almost the same value. So this is something that is, we think is quite important, but there's still a lot of research to be done really in soil. 5 minutes, good, thanks. And just more recently or so, this was some work proposed by Danny, the colleague Gang Wang. They thought, oh, can we use those concepts to propose something like a coexistence index? So they thought, oh, we could come up with something simple. We'll look at again, we're going to use the metric potential as really this driver of the processes here. We're going to look at, under wet conditions, what is the ratio between the mean generation length, that is the distance a cell can travel during a generation time, and the RC here, the size of the largest connected cluster. So a connected cluster just means that you're in a soil environment, and it is wet enough that you can go, if you're a cell, you can go from point A to point B. Doesn't mean that if you're blocked, doesn't mean there's no water, but at least the film is too thin for you to go through. So under wet conditions, you have a few colors. That means there are a few large domains. And as you dry, you have many small domains. So if you're trapped in one domain, you cannot escape. And they use this to compute this coexistence index and compare with some of the data that I just showed you previously on these sand microcosms. And we find some elements that tend to support this, that under dry condition, you get both species, you get coexistence of the 2 species, and it's not the case when it's wet. So of course, a lot of processes, a lot of elements are not present in such a very simple way of representing the problem, but we think that it's a possibility that it can, we can use such idea to try to model a little bit better and predict a little bit better what happens to microbial diversity and interactions in porous media, including in soil. With this, I will, I think for the sake of time, I will skip the last part, but it will be also present in the PDF. So I had a few more slides, but I'm going to skip them. I'm going to jump to the take-home messages. So really, I hope I convince you about that really the main player here for microbes is the soil aqueous phase. So we saw the importance of the soil aqueous phase and especially metric potential for motility and dispersion, spatial organization, respiration, coexistence, and that this soil aqueous phase is a unifying control over activity and the different processes. What I want to stress as well is that the soil water influence microbiology at all scales. You've seen microscopic elements, you've seen also larger scales. You can really think of it at all of the scales that are relevant to your research. I just added finally a little micrograph of fluorescent bacteria. Again, the Corsican bacteria that we isolated here, where we label them with different fluorescent dyes.
Because you showed also unsaturated media?
Yeah.
But I'm also talking specifically on saturated media.
Yeah. So I will give you the favorite answer of the biologist, which is it depends. I mean, I really don't know like this. I mean, I'm sure that some of the processes that you mentioned will happen if there's preferential flow. That obviously the availability of nutrients will be a big driver. If there's not enough nutrients, there won't be growth or there won't be clogging. So depending on if we're talking about an artificial system where there's nutrients that are constantly brought in, there's a much higher risk of getting those kind of clogging, I believe. Maybe in the subsurface, you don't have enough growth or sustained growth for that to take place. So for that reason, I say it depends. Then if we look specifically at some cases, we could maybe suggest things, but without more context, it would be a bit difficult.
Can they grow also upon each other or only upon films of the surface?
So the question is if the cells can grow on top of each other. Well, certain species, yeah, absolutely. I didn't show that, but you can have so-called biofilm growth or colony growth where you really have hundreds, thousands of cells that pile on each other. The problem for them is if they grow on a surface, depends where the nutrients come from. If the nutrient is all around them, that might not be a problem. If they are growing on a piece of organic matter, let's say, and they have to acquire the nutrients from it, if you're on top of the biofilm, then you may not get much because there's no way. It's all the diffusion. All the molecules are already intercepted before they reach you by the other cells there. So it really depends where the nutrients come from, and you can have relatively complex structures, 3D structures, at the micrometer to millimeter scale, depending on the situation.
Let's yes, very quickly. So for a typical soil, what would be the volume fraction of bacteria? I sort of thought it was sort of 0.1% from what you showed.
Yeah. But it was not. It's not volume. It was the surface coverage. The volume. Well, usually the thing is that you could think of the highest number for bacteria. So if we think of very rich soil, we might say, okay, there's maybe 10 to the 10 cells per gram. So then we could try to do a little bit of math to see what that would mean in terms of of surface coverage or water for the volume. Then I don't know, what's the amount of water that we can store in 1 gram? But then we can calculate what would be the density. But in most cases, you could imagine that you'd probably still pack more of those. It's not that that the medium is so busy, is so full of bacteria that you cannot put any more. Usually, you would still, one explanation for this is that you can add some extra nutrients, add some glucose, add some other things, and you're going to get some growth. So clearly, they still have room to expand. Usually, it's because they don't have enough resources.
And in terms of specific surface area, if you compare to the soil, is it sort of significant, or is it just 5th digit or something like this?
Well, the values showed saying that it's much less than 1%. Comes from different estimates. Where, indeed, also in terms of volume, they think that it's really relatively small. I mean, it might be 0.01% or something like this. Depending, then that might vary depending on the soil texture and whether all the surfaces are available to the cells or not. There might be big differences. Yeah, probably.
I'm just curious. At the beginning, you mentioned that soil bacteria or many soil bacteria are used for vaccines.
No, sorry. What I said was that most of the antibiotics that are used today were first originating from soil bacteria.
Is there any reason for that?
Well, the reason is that bacteria are the ones that are interested in developing both antibiotics and antibiotic resistance because they use it. So for a long time we thought they just use it as a warfare. So they use to kill others. So you kill your neighbor, you can take its food. That's the way. But now we have a more refined view. We think that they can use also antibiotics to communicate, to signal certain things. But the bottom line is that really antibiotics and antibiotic resistance, which is a huge problem today, are very ancient. So you can go in soils that are thousands of years old, and you can that were never in contact with any human activity, and you can isolate antibiotic genes and resistant genes. So that's part of their ecology. They use those sometimes to maybe kill neighbors, sometimes to communicate, and it's really out there a lot in soil.
I understood correctly, it seemed in one of the slides that the soil water potential affected the microbial respiration strongly, even when it was near saturation, like when the saturation was near 1. Is that correct? Or?
So what I showed was that you had the soil on the one axis. You had the soil water potential. The other one was soil relative humidity. And the soil relative humidity across a long range of water potential stayed close to 100%. But what it showed is that if you want to know how microbial activity and functions are affected, then you want to look at the potential, because that's clearly what will affect them, the relative humidity in cell.
How do you explain that? I mean, the soil is almost saturated, but the microbes...
Yeah. That's a very good point. The way we understand this is that it makes a huge difference when you change potential. What is the gas and water phase distribution in soil? Because imagine that, well, we saw for dispersal that can limit dispersal, but then you have this way increased of surfaces that can then be direct exchange with the gas phase, and that could be really a huge difference. For that reason, it's relatively, it's difficult to reproduce in the lab exactly those in an artificial system the effect of the metric potential, because you can use chemicals to mimic that, but you don't get the gas aqueous phase effect, and this is really the key thing. If you don't have this effect, then you lose probably the main mechanism that is changing their activity.
You've talked a lot about cells.
Yes.
But the cells put out a lot, surprisingly. But the cells put out a lot of stuff, polysaccharide, other materials that affect the surface tension and the physics of the water around them.
Yeah.
And sometimes are a much larger volume than the cells themselves. Can you talk
Absolutely.
a little bit about the extracellular materials?
Yes. No. Sure. Very quickly, I will show you the slide that I skipped. Sorry for the sake of time. Yes, cells can also produce extracellular material. You see here a cell, and there's this whole material here is so-called extracellular polymeric substances, which is a mix of polysaccharides, of all sorts of things. And this has an extra high capacity to read in water, and this, of course, will change both the biology of the cell, but also the microstructure of the soil. And here, just quickly. Here are some experiments that were done about water retention on sand, simple experiments. Sand supplemented or not with EPS. You have the water potential here, water content. So what you see is that when you add EPS, you can vastly increase the water content here compared to sand that doesn't have EPS. Another thing is that EPS can lead to hydraulic decoupling. Those were experiments made in the glass beads column. You see the blue, it means that it's saturated with liquid. So you first saturate your column, and you add a little bit of EPS that was added to the column. And after drainage, what you see is that the blue tend to stay where the EPS was. So you have this retention of the water due to the EPS. So it's a very interesting component of the soil, which is like this interface between the biology and between the soil physics. And it's really an area with a lot of interesting research, but we're still far from getting the full picture of what role it plays exactly in all porous media. But it's definitely a very interesting topic, which is making the link between the soil biotic and abiotic components.
Yeah, I have a question. Is there a depth limit to which you can actually observe microorganisms, and is this controlled by the pressure, the porosity, and temperature? And I mean, worked in Greenville at 500 meters below ground surface.
if I understand you correctly, what you say, is there a depth limit to observe micro?
Across like at 1, 2 kilometers, like extremophiles and things like this.
Oh, extremophiles. So, microorganisms that you think should not survive in their environment but do survive. Well, okay, to my knowledge, I mean, even if you think that nothing should grow, no microbes should survive in any given environment, we always tend out to find them, whatever happens. In the deepest sea environments to the hottest environment, you find microorganisms that survive high radioactivity in nuclear plants. It seems that as long as you try hard enough, you will find them anywhere. Maybe just not very abundant, but you can find really microbes that survive at boiling temperature, survive high dose of radiation. So it's very difficult to say there's no microbes that are in these environments. People will tell you it's because you've not searched hard enough. Yeah. Now that's a very good question, and actually there's a PhD student in our group who's trying to do this to try to connect the global scale and those kind of phenomena we're talking about. So looking at in different biomes or area, what is the frequency of rain events, and what is the environment? How could we use this to model and predict what may be, for instance, the genetic exchange leading to some evolutionary changes over longer periods of time. We've done that also on a smaller scale, looking at direct gene exchange between bacteria and porous media as function of matrix potential, and we do find some effects. So there is some attempts. We're still not clear. It's not the full picture. We're far from the full picture. It's really small steps. But we do think that there's a way to connect the larger scales to the microscopic processes, and trying to then better predict in a given environment what we should expect in terms of gene exchange, evolution, and whatnot.